So those of you who do not know me, my name is Rob Turesky. I'm a professor in the Department of Medicinal Chemistry. I conduct basic research on chemical carcinogenesis in the Masonic Cancer Center and the Cancer Chemo Prevention Program. So my lab is very interested in chemicals that we think are cancer-causing agents. I look at a number of different types of chemicals, and I will be using two classes of carcinogens and how we approach biomonitoring as prototypes today. Okay, so we're exposed to many chemicals in the diet and the environment through, for instance, emissions and exhaust gas. We all know that tobacco smoke is very hazardous for our health. There are mycotoxins such as aflatoxin in corn, in rice, and other staples, and Aristolochic acids, which are present in the herb genus Aristolochia, which is actually used in traditional herbal medicines worldwide. And also when we cook or flame broil meats, we produce a series of carcinogens. Some of these chemicals we think contribute to cancer risks. So my outline of my talk today will be on two classes of carcinogens, including the heterocyclic amines, which are formed in cooked meats, bacon, poultry. Some of them also arise in tobacco smoke. I'll be focusing most of my talk on the HAA called PhIP, which we believe contributes to prostate cancer, pancreatic cancer, colorectal cancer. These are target organs in animal models, and based upon epidemiology studies, individuals who eat well-done cooked meat are at elevated risk for these types of cancers, and PhIP may be a causative agent. So I'll talk about some of the biomarkers of exposure and DNA damage and biological effects that we've been studying on PhIP. And then I'd like to continue on and discuss some of the work in Aristolochic acid, which we've used, which is a upper urothelial tract carcinogen, renal cell carcinogen, bladder carcinogen, and liver carcinogen, again, that occurs in some traditional herbs that are used worldwide. Time permits, I'll talk about some of our novel approaches to measure DNA adducts in paraffin-embedded tissues. This is a breakthrough technology. And then I will present some of our developing research on DNA adductomics, screening the genome for the totality of DNA damage. It's clear that in epidemiology studies, there is a gaping hole in the implementation of validated measurements of environmental carcinogens and their biomarkers. And with the advancements in mass spectrometry techniques, there's a tremendous opportunity for us to better understand the chemicals in the diet environment. They may be contributing to disease risk, including cancer. So traditionally, epidemiology was like a black box. We understood lifestyle factors, external exposures, and there was a disease outcome, but we really don't or did not know what some of the causative agents were for these diseases. Over the past 2 decades, there's been an explosion in genetic biomarkers, genomics, genetic polymorphisms, the ability to link those to specific chemical exposures and DNA damage, though, have lagged. So we can measure chemical exposures. For instance, we know what's present in tobacco or what's present in grilled meat. We can estimate the internal dose by measuring some of these chemicals or their metabolites, for instance, in urine, blood. Some of them will bioaccumulate in hair or fingernails. This is the internal dose. In the case of many carcinogens, they need to be metabolized to reactive intermediates that will covalently modify proteins or DNA. This type of biomarker would be termed the biologically effective dose, the percent of the dose that is converted to the reactive intermediate. And then there are multiple biological effects that need to occur in order for a cell to become cancerous. Ultimately, what we would like to do is we would like to be able to link specific DNA adducts with cancer, with mutation spectra in cancer driver genes to further strengthen the association between the chemical exposure, the DNA adduct, with mutations. This is an example of the cooked meat mutagen PhIP, which gets absorbed in the body and gets metabolized in the cell. There are multiple pathways of metabolism, and the major pathway of bioactivation occurs in the liver through N-oxidation to form the N-hydroxy metabolite. This is a reactive intermediate that can bind to DNA, and fortunately, we have repair systems in our bodies that can repair most of these DNA lesions. However, some lesions may escape repair, and during cell division, this DNA adduct can induce a mutation, again, through multiple processes, produce a cancerous cell. Depending upon the level of DNA damage, you could actually get cell death or apoptosis. In addition, some compounds, including PhIP, we believe induces oxidative stress. In this case, PhIP can bind to the estrogen receptor or the androgen receptor, stimulate cell proliferation, oxidative stress, which can induce endogenous electrophiles, which can further damage protein or DNA, and also contribute to cell proliferation and toxicity. So chemical biomarkers represent recent exposures, while the mutations are an accumulation of exposures over the lifetime. So DNA addicts represent internal dosimeters of exposure to genotoxicants. They have been used for many years for interspecies extrapolation of toxicity data in rodents for human risk. They can be used for individual susceptibilities. We all have different genetic makeups. We have different levels of expression of various enzymes that are involved in metabolism and DNA repair that are going to affect the biological potency of certain procarcinogens. Some DNA addicts have been used and are being used in chemoprevention, and also chemotherapeutic drugs which act by forming certain types of DNA addicts or cross-links. The measurements of these addicts can be used in precision medicine protocols to assess efficacy. Identification of DNA addicts and their mutagenic potential among the first steps in assessing the role of a chemical in human cancer etiology. And some DNA addicts are a critical aid in formulating public health policy that can aid to reduce chemical exposures and hopefully decrease cancer incidence. So again, I'm going to talk about PhIP in cooked meat. The IARC, International Agency for Research on Cancer, which I served in this working group several years ago, classified processed meat as probably carcinogenic to humans, and they were focusing on colorectal cancer. Processed meat, salami, sausages, bacon, and red meats, beef, veal, lamb, is also been linked to colorectal cancer. It's designated as group 2A, signifying there's very strong mechanistic evidence and in epidemiological associations. It's just a little bit weaker than the data for the group 1 processed meat, which is clearly defined as carcinogenic to humans. In addition, red meat is also positively associated with pancreatic and prostate cancer based upon a compilation of epidemiology studies. There's more than 25 genotoxic HAAs that are formed in cooked red meats. Some of them also arise in tobacco smoke. Again, most of my talk today will be focusing on PhIP because it is the only chemical mutagen formed in the diet in cooked meats that is known to induce prostate cancer, which I'll address. It also, as I said, it's a multi-site carcinogen in rodents and induces colorectal cancer and pancreatic cancer as well. There are nitrosyl compounds formed in preserved processed meats. I will not be discussing these classes of chemicals today. So this is kind of a cartoon that explains how we get exposed to these compounds. So if we're cooking, barbecuing, grilling high temperature meats, we will produce a series of carcinogenic compounds at low levels, parts per billion levels in general. And again, this is the PhIP molecule that we're discussing today. It gets absorbed in our body after consumption. It gets metabolized primarily in the liver, and its metabolites can undergo systemic circulation to various target tissues such as the prostate, the colorectum. There is some limited epidemiology data suggesting cooked red meat contributes to bladder cancer risk, so it's possible maybe this compound could contribute to bladder cancer. In any event, we believe most of the metabolism and the genotoxic metabolites are formed in the liver, survive, go through the bloodstream, and undergo further metabolism and DNA damage in these extrahepatic tissues. Of course, we need biopsy specimens to measure these types of damages, and that's not so easy to obtain, obviously, because surgeons are very reluctant to provide tissues for basic research just because the tissues are very scarce. So we look at alternative methods to try to assess exposure and potential damage. In the case of PhIP, we discovered, well, we didn't discover, we've adapted methods in the literature where we've actually seen that PhIP in particular, which has high affinity for melanin, as the PhIP, unmetabolized PhIP goes through the bloodstream, it interacts in the follicle with the developing melanocytes and non-covalently binds, and as the hair shaft grows out, the carcinogen's trapped in the cortex, and you can snip a little piece of hair, and you can measure for an individual to see if he or she was exposed to this carcinogen via the diet. We can also measure compound PhIP or its metabolites in blood, although that's transient. Some of the reactive intermediates can covalently bind to proteins such as serum albumin, and I will briefly discuss some of our data there. And we can characterize the metabolome of PhIP by looking at the metabolites in urine. Many of these are detoxified metabolites, but also conjugates, semi-stable conjugates of the carcinogenic metabolite. And so we can understand how people biotransform this compound differently. So how do we do this? We do this by mass spectrometry methods. We need very sensitive, and I might add expensive equipment in order to measure these compounds or their biomarkers at trace levels. We get low quantities of biospecimens. We need super sensitive analytical methods to measure these. We like to be able to conduct absolute or semi-quantification, and it's really important to get precise structural identification to make sure that we're not getting false positives. And again, we can measure compounds in fingernails, blood, hair, and exfoliated cells in urine, for example. So what we did is we, as part of an NIH-funded grant with the University of Hawaii, we developed a semi-controlled feeding study where we had a group of healthy young adults, graduate students for the most part in Hawaii, who ate well-done cooked beef for 5 days a week for 4 weeks. And we had a pre-feeding stage for 3 weeks where we asked them to refrain from eating any putative types of foods that would contain these genotoxicants in well-done cooked meat. And then we had a 4-week feeding study, and then we had a washout period for another 4 weeks so we could look at the decline of these biomarkers. And we collected newly grown hair at the end of the pre-feeding, after the post-feeding, after the feeding, and then another 4 weeks after the post-feeding. And we collected blood drawn at these different time points as well, and also urine. And so the first biomarker I'd like to talk about is the albumin adduct. Albumin is the major plasma protein. It has a number of biological functions. It's a very stable protein, in part because it has 35 cysteine residues, of which 34 of them are tied up as 17 disulfide. So it's a flexible molecule, but it's quite stable. And there is one sole cysteine, the cysteine 34, which is in an acidic microenvironment in a small cavity of the protein. And it's been shown mostly in vitro that a number of reactive xenobiotics, electrophiles, can get into that cavity and can react to that cysteine 34 to form DNA adducts. And you can see there's on diclofenac, acetaminophen, oxidized quinones, estradiol, acrylamide have formed adducts there. And we characterized the adduct of PhIP formed in vitro, and I will also show you some of the data that it forms in vivo. So the biochemistry and metabolism is such as, again, the PhIP undergoes N-oxidation to form the hydroxylamine in the liver. It can go through the bloodstream, or in fact, it could occur in the liver. It can undergo, it's a reducing agent. It can get oxidized to the nitroso intermediate, and the nitrosas are very selective substrates for thiol groups. And in our case, we've characterized this sulfonamide that forms in vitro. And what happens, so what we do is with these subjects, we collected small amounts of their blood. We need about 1 milliliter of blood to do this assay. We separate out the plasma from the erythrocytes by centrifugation. We use an affinity blue dye, which is very selective in pulling out the albumin, very high level of purity in plasma. We treat with mild acidic pH so that the albumin itself remains intact, but we cleave this sulfonamide linkage to regenerate the parent amine PhIP and the sulfonic acid. And interestingly, the biochemistry for 2 other prototype HAAs, these chemicals form this adduct in vitro when you use the genotoxic and hydroxy metabolites, but we haven't been able to identify them in vivo. But we have been able to indirectly measure PhIP. So we isolate the PhIP by a liquid-liquid extraction followed by mixed cation exchange and do quantitative LC mass spec. And what we saw in this group of healthy young adults is that we saw low levels. We had 2 groups here. We had low levels of PhIP detected in the pre-meat feeding stage, but then at day 21, we had them on for 4 weeks, and we're getting a significant jump up in the level of the adduct. And then we had them cease eating well-done meat at day 49, and you can see there's a rapid drop-off. 2 weeks later, greater than 95% of the adduct has been removed. Same phenomenon features occurred in this group, which is done a semester later. And so this data shows us that PhIP does indeed undergo bioactivation in vivo. This is the first report of that. So it is being bioactivated to a genotoxic metabolite. We have a very large spread in the range of the adduct in these subjects, over 500-fold. I mean, that's a tremendous spread. Interestingly, this was not reflected by the urinary biomarkers of this metabolite, which only varied by 2 to 3-fold. We worked very hard in developing this biomarker. It's research that's taken us 3 to 4 years to do. Unfortunately, the adduct, the half-life is very short. We've estimated it's only about 3 days, while that of albumin is over 21 days, so that linkage is not super stable, which tells us we probably cannot employ this as a robust biomarker in subjects in epidemiology studies of individuals who are eating a free choice diet. The take-home message is this compound undergoes bioactivation in vivo. Let me go to the hair biomarkers. I mentioned in the introduction, a small amount of the PhIP after first metabolism circulates through the blood system and interacts with the melanocytes and the growing hair follicle. And as that grows out, the carcinogen rendered as these yellow balls, it's in the cortex. We can take a small snippet of hair and we can measure for it. And again, I'm an analytical chemist, mass spectrometrist, so I like to show some structures and mass spectra. Anecdotally, some of the epidemiologists had thought that individuals who had darker pigmented hair had higher levels of PhIP than those individuals who, for instance, had gray hair or light blonde hair or red hair. When I was developing this assay, we do not have vegetarians as subjects, as negative controls, because PhIP only occurs in cooked meat. So vegetarians would expect to be negative. This is my beautiful dog, Moses. And he actually helped me with this experiment. Actually, I thought we were just going to take his fur as a negative control because we don't give him grilled meats or steaks or scraps. And what we did, I was very surprised to find out that he had a lot of PhIP in his fur. It was only in his black fur and not his white fur. That proved actually that pigmentation is very important in the accumulation of this carcinogen. It turns out that in a study, we followed up with this with Jamie Mondiano at the vet school here at UMin. We looked at a number of dogs and almost all of them were positive. And it turns out that most of these dogs, they were eating kibble, high temperature, dried, shelf life food for dogs. And many of the kibble contained poultry. It's a very inexpensive source of protein that's commonly used in canine kibble. And we know from our data and those of others that actually poultry at high temperature produces very high levels of this carcinogen. So we don't give our dog any more kibble, particularly that containing poultry, because it's possible this this chemical could be contributing to canine cancer. I'd love to do more studies on this. It's been a challenge to get funding, however. So take-home message is if you have a dog or perhaps other pets that are eating kibble with poultry, I'd strongly advise you to change their diet. So going back to this Hawaiian cohort study, what we did is we had individuals eat meat at different levels of doneness of meat, and we estimated how much of the PhIP was in their hamburgers on a daily basis. So we had individuals that ate about 400 nanograms per day, which is the average intake, we believe, to more well-done meat. There's some scatter, but you can see we get a nice dose-dependent increase in the levels of PhIP in hair as a function of how much they ate in the diet. You can see at the highest dose, this almost looks like an outlier. But as I told you, the compound has high affinity for melanin. And if we normalize our data for melanin, you can see we have a much better curve here. And so it's very important to normalize the PhIP levels for melanin. Interestingly, many of the other prototypical HAAs, they have much lower affinity for melanin, and it's difficult to actually measure these other heterocyclic amines in air. So metabolism of PhIP, it's actually quite complex. We tried to associate biomarkers, and we haven't been able to get some of the correlations that we had expected at the onset. So again, PhIP undergoes N-oxidation by CYP1A2 in the liver to form the hydroxylamine. This has a number of metabolic fates. It can undergo glucuronidation to form these two isomers. Again, these are conjugates of the carcinogenic metabolite. We tried to correlate this to caffeine phenotyping in these subjects in Hawaii, and these are biomarkers of metabolism of CYP1A2. We expected to see a positive correlation. We did not. We saw no correlation. We expected to see an inverse correlation with PhIP in hair. We did not. We would have anticipated that the rapid CYP1A2 phenotype would have higher levels of hydroxylamine in blood, and we'd form higher levels of the sulfonamide adduct, the albumin. We did not. And it's difficult for us to make this correlation with DNA because we just don't have enough subjects. But the metabolism of PhIP gets quite complex. There are reductases that will reduce the hydroxylamine back to the parent compound PhIP. NADH reductase, glutathione reductases. This may be masking the CYP1A2 phenotype. Not only that, but this enzyme is highly inducible. The coefficient of variation per individual over the course of the 4-week feeding study varied by over 100% from week to week. So it's highly variable. So unfortunately, this metabolic phenotype is not robust, and we can't use it for correlating with our different biomarkers. Well-done cooked meat has been linked to aggressive prostate cancer. The epidemiology data, however, on dietary factors and prostate cancer risk are inconsistent. We wanted to know whether some of the biomarkers of DNA damage could help us to understand chemical agents in the diet that contribute to prostate cancer and cancer risk. And again, this is work that was done here at the University of Minnesota with the Department of Urology. And I need to thank Dr. Chris Waite and Perry Murigan for all the wonderful support and collaborations they've done with us on this project. So this is 5 different carcinogens that are present in the diet or environment in cooked meats. And we measured DNA damage in the prostate by high resolution mass spectrometry. Our LOQ, our limited quantification, is very low, 2 addicts and 10 to the 9 bases. We've gone through about 100 patients, and the only addicts we've seen thus far are those of PhIP, not these other carcinogens. Let me talk to you about measurement of DNA addicts. We do this by ion trap or Orbitrap mass spectrometry. First of all, in order to have a validated method, you must be assured that the conditions used to isolate the DNA preserves the structures of the addicts, that they don't decompose. We have to digest the DNA down to the deoxynucleosides. We must make sure that the addicts don't interfere with the enzymes for this digestion. And again, we need mass spectrometry to unequivocally measure and quantify these. So we do this by mass spectrometry at the MS to the 3rd scan stage with the ion trap. So basically what you have is you have your DNA addict, you run it through an HPLC into the mass spectrometer, and we do what's called tandem mass spectrometry. We fragment, we break the molecule. The most labile bond is that of the sugar, the deoxyribose, and we get the A-glycone base. And this actually gives us a single fragment, which is a nice cleavage, but it gives us limited data. But the way the ion trap works, we can collect this fragment ion and we can further break it apart a second time, and we can get this nice fragmentation pattern or fingerprint to corroborate the structure of the adduct. And this is an example of a cancer patient. This is a subject who was negative. We have an internal standard that we synthesize, so it has the same chemical properties. It's just got a delta 10 mass unit. And this was an individual who was positive. And when we do MS to the third, again, we get this beautiful fragmentation pattern and corroborates the structure of the adduct. We've gone through actually 100 patients, 110, 120 patients thus far, and we get about 10% positivity. We see similar levels of adducts in the peripheral and transition zones. So this is consistent with some of the epidemiology data that well-done cooked meat and perhaps PhIP may be contributing to prostate cancer. One of the challenges we've had, and we've only discovered this by measuring the PhIP in hair, our group of cancer patients where we also collect their hair, the levels of PhIP in their hair, although this seems like it's a lot, it's actually not. The limit of quantification is 25 parts per trillion, and our median is just above that, and our mean is only twofold above that. If we plot this as a histogram, those healthy subjects in Hawaii, their levels of PhIP in hair were out about here. In contrast, we're very much skewed to the left. It indicates, even with normalization for melanin content, it indicates our cohort is not eating a lot of meat. We do have some subjects here, however, who are eating quite a bit. And again, this is a scatter plot showing that for the most part, we don't have a high frequency in this cohort. So what we really need to do is go to another cohort where there's more frequent consumption of meat and to see if we're getting more positivity. But even so, I think this is significant. About 10% positivity is not negligible, and it does affirm that PhIP is damaging prostate genome DNA. We've recently looked for other markers of DNA damage, including oxidative stress. And what we've looked for a number of lipid peroxidation adducts in prostate. And what we've seen is actually 6 and 8 propano are frequently detected. And this is, I just plotted this last night. I don't like the way I plotted it, but the point is there are a number of patients that are harboring these adducts. We'd like to try to see if we can correlate these biomarkers, these adducts, to Gleason score and other pathologies to see whether we can determine whether these might be useful biomarkers in the future for prognosis or aggressiveness of the cancer. We have looked at PhIP in hair, as I mentioned on the other slide, and this histogram is deceiving. We have about less than 10% of the cohort that has levels 10 times above the LOQ. We are actually asking the question whether the subjects with high levels of PhIP in their hair, we know it doesn't correlate to DNA damage because we just don't have enough subjects, but we are looking whether there's a correlation with a Gleason score and tumor aggressiveness. We'd like to go back and look and see if there are other chemicals in here that could be linked to prostate cancer or other diseases. We have followed this up with some in vitro work in cell lines, LNCAP cells, prostate cancer cell lines. We looked at the parent compounds of PhIP and 3 other prototypes. You can see is that they are non-toxic in these cells, which makes sense because the cells do not express CYP1A2, which is required to bioactivate them. But if we incubate the cells with the genotoxic synthetic metabolites, what you can see is PhIP is the only one of the 4 that is inducing toxicity in a dose-dependent manner. We've also looked for DNA adducts of these carcinogens in the cell lines, and consistent with the toxicity data, PhIP is the only heterocyclic amine that's forming high levels of adducts. Moreover, we've done a dose response in these cell lines, and we can see it's nice dose dependent, and we can measure adducts in these cell lines at levels that are approaching human exposure. So this recapitulates some of our in vivo data that of the HAAs, PhIP seems to be the major player in damaging DNA of the prostate. We know that cancer is multifactorial. Clearly, there are many subjects, individuals who are developing prostate cancer, and PhIP is not the only driving agent for this. And not only that, but PhIP in itself could be contributing to the prostate cell cancer in different ways. We know it does bind to the androgen receptor, which could have an impact in cell proliferation, oxidative stress, and inflammation. And it's known certainly that environmental exposures can affect transcriptome, thus the proteome, and the metabolome. And we have some preliminary data on PhIP that I'd like to share with you. We incubated PhIP, or its genotoxic metabolite, and we looked for cytotoxicity and cell growth. What we have in here are our control cells. We were just incubated with DMSO. If we treated the cells with 10 nanomolar dihydrotestosterone, which is a ligand, a natural endogenous ligand for the androgen receptor, you can see we're getting strong induction of cell growth. PhIP itself did nothing. Again, the compound's not well metabolized in the cells. N-hydroxy-MEIQX, a genotoxic metabolite of another HAA, also really did nothing, but N-hydroxy-PhIP did not induce toxicity, but it arrested cell growth. Phenotypically, these cells appeared to be in a senescent state, and we believe that's because of the adducts that are formed in the cells that are preventing the cell from dividing, so they're more or less in a quiescent state. We've looked at the impact of PhIP on certain metabolites through metabolomics, and what we saw is that when we treated with N-hydroxy-PhIP, there was a dose-dependent decrease in glutamic acid, approaching that of butyl hydroperoxide, which is a very potent oxidant. We also saw a dose-dependent decrease in glutathione, a key antioxidant and chemoprotective agent in the cells. So that indicates that oxidative stress is occurring by PhIP, and we always back this up by mass spectral data to support the identities of these analytes. Again, we have perfect matches with reference libraries. We also discovered during the unbiased metabolomics approach that PhIP actually decreased the levels of S-adenosylmethionine, or SAM, which is very exciting for us. SAM is involved in many different biological effects within the cells. It methylates DNA protein and is contributing to epigenetic events. It's also a key intermediate in glutathione synthesis. So again, our data indicates that PhIP is inducing oxidative stress, which is another contributor to prostate cancer risk. So in summary, PhIP is detected in hair of omnivores. About 50% of our cohort has low levels in the hair. We're looking at the correlation of PhIP hair levels with prostate cancer and pathology. Can the hair exposome be used to screen for other carcinogens and these risks? I'd like to do research on this. We know that PhIP undergoes bioactivation in humans. The PhIP-DNA adduct is sporadically detected in the prostate genome. We don't know anything about the capacity for DNA repair. One of the challenges is that we do not have characteristic mutations of PhIP and cancer driver genes. Are epigenetic events and oxidative stress induced by PhIP in the prostate? We'd like to do more research on this. And there are many, many other HAAs formed in cooked done meat. We don't know what their role is. There's a synergistic role, and we hope to do further work in this area. So let me switch over to Aristolochic acid. This is a completely different chemical. It is produced naturally in the herb genus Aristolochia, and there are two major cohorts in the world. The Balkans have high levels of exposure to this carcinogen, particularly in small rural villages which still produce their bread in traditional practices, where the herb Aristolochia unfortunately is commingling with the wheat fields, and when the wheat is cut, unfortunately the herbs are being cut with it, and this carcinogen it occurs in the bread that's being used in these rural villages. And in Asia, China and Taiwan, this compound unfortunately is present in many traditional herbs that are used in medicines. AA exposure occurs worldwide. Despite FDA banning, you can still unfortunately detect Aristolochic acid in products that are sold over the internet in the USA. It causes rare urothelial carcinomas of the upper urinary tract, and as a signature mutation, it's an uncommon AT to TA transversion in the P53 tumor suppressor gene, but it also occurs in other cancer driver genes, and we think it's attributed to AA. And it is a group 1 carcinogen by IARC in 2002. This is an outdated slide. We became aware of Aristolochic acid in 1993, where a group of women in Belgium were exposed to this carcinogen in a slimming regime, and they developed renal failure or renal cancer. The exposure risk in the Balkans is thought to be 100,000 people. That in China, it's over 100 million who use products containing this carcinogen. In Taiwan, over 8 million people at risk. Again, this is the carcinogen, the DNA adduct of the carcinogen that we believe induces these mutations. Most of the work done worldwide has been done by post-labeling monitoring. I believe our lab is still the sole lab in the world that still routinely measures this carcinogen by mass spectrometry methods. Again, the adduct induces a rare mutation. This is work by Arthur Grolman at Stony Brook. What he did is he looked at the P53 mutations in the Balkans and in Taiwan on the coding region of P53. And what you can see is that these two distinct parts of the world, the mutations, they're almost superimposable. I mean, this just does not happen. And if we look at the percent of mutations, the AT to TA mutations in these two cohorts, they're very prominent, 50 to 65%. In contrast, the IARC database worldwide, it's only several percent. So that's highly suggested that these two cohorts are exposed to the same carcinogenic agents. We've developed biomarkers. We have the DNA adducts. There's mutational spectra. There are mutational signatures that are being used to assess exposure. This is some of our work. This is an individual, we have internal standards here. This is individual renal cortex where we cannot detect the adduct, and this is our limit of quantification is very low. This subject's very high, and again, we have the product ion spectra to corroborate the structure of the adduct. This is a cohort in Taiwan that we've analyzed. In contrast to PhIP, you can see this cohort has very high frequency of this DNA adduct. This really, it doesn't happen. 90% positivities for an adduct. It's incredible. Taiwan has the highest levels of upper urothelial cancer in the world, and they also heavily use this traditional herbs, unfortunately containing this carcinogen. We've looked at several other cohorts. Again, in Shanghai, this is a different split scale, so please note the difference. But even so, we have appreciable positivities in Shanghai, the Balkans. We have lower numbers in Japan and South Korea, but clearly this carcinogen is prevalent worldwide. Ghislaine Scalo at IARC did a study in Europe several years ago where she looked at 4 different cohorts: the Czech Republic, Russia, United Kingdom, and Romania. And what she had discovered is that as the number of somatic mutations increased in these different cohorts, it was only the cohort in Romania where the AT to TA mutations increased. She suspected it could be due to Aristolochic acid. She came to us and we did this blind coded. We found positivity only with the cohort in Romania, not with UK, Russia, or Czech. We did this blind coded, and I was quite pleased by this. And as the log of the adducts increased, the proportion of AT to TA mutations increased as well, reinforcing the notion that Aristolochic acid is contributing not only to upper urothelial cell cancer, but to renal cell cancer as well. We have done a little bit of work on exfoliated cells. This is the little piddler in I think it's Luxembourg, a very famous statue. Every day when we urinate, we do slough off cells from our renal system and our bladder. It's not a lot of DNA, and this is not a high throughput method, but this is sensitive. This amount of DNA is sensitive enough for us to assay for DNA adducts and Aristolochic acid. And there was a cohort that we collaborated with in Beijing with subjects who had ingested Aristolochic acid through the herbs. This is one subject, his urine was collected 24 hours after he ingested the herb. Tremendously high level of the adduct and exfoliated cells in urine. The subject was assayed again 6 months later, a drop, but still measurable. And there were other subjects who claimed they had stopped ingesting herbs containing this carcinogen over 8 years ago. This adduct is known to be extremely persistent. It doesn't undergo NER repair. And so this is potentially a very nice non-invasive biomarker to screen for cohorts in Greater China and Taiwan non-invasively. We don't need a biopsy specimen to do this type of work. So that's all fresh frozen work. Oftentimes we cannot have available fresh frozen tissues, but formalin-fixed paraffin-embedded tissues are available for, with clinical diagnosis, and we have asked the question whether we can assess DNA damage in formalin-fixed tissues. Because of the processing of formalin, preservation of formalin-fixed tissues, it's been extremely difficult to measure adducts. However, we have developed a method that allows us to do this. Again, formalin-fixed tissues are the most commonly used preservative worldwide. You get extensive cross-linking of the tissue, and then the tissue undergoes dehydration, and then it's placed in hot wax, melted, and you have a section block. The pathologists take thin wafer section cuts, and they can do cell histology or immunohistochemistry. However, the condition used to reverse protein DNA crosslinks or DNA-DNA crosslinks have traditionally required elevated temperature and alkaline pH. That will damage most DNA adducts. And so for us to be able to be successful, we need a method under mild conditions, mild pH and temperature, that will completely reverse the crosslinks and allow us to measure the adducts. And we were funded to do such work, and I'll show you the technology in an instant. But basically, this represents 11 subjects from the Balkans where we had matching fresh frozen tissue and formalin-fixed tissues, some that were stored for up to 9 years in a drawer at ambient temperature, and we were able to retrieve the bulk of the DNA adduct. So this is a very exciting technology. And originally we adapted a method by Zymo manual spin kit. They have proprietary reagents that are reversing the crosslink under mild conditions. We've adapted this now to a semi-automated method, which lets us assay more numbers of samples per hour, making it a larger high throughput assay. We've also looked at other classes of carcinogen adducts. So in summary, our work in Aristolochic acid exposure occurs throughout the Balkans and Asia, but it also occurs in other parts of the world. There are herbal medicines still contain this carcinogen. Mutational data shows the contributions of Aristolochic acid to upper urothelial cancer, but also renal cell, bladder, and liver cancers. With an inexpensive ion trap, we'll be able to measure this exposure through DNA adducts. FFP tissues are untapped specimens that we can use to measure adducts of Aristolochic acid and other carcinogens. And at least certainly in the case of Aristolochic acid, it's been correlated to mutational spectra. We have been able to apply this technology, for instance, to NNK, tobacco-specific carcinogen, benzopyrene as well in animal models. We expect that this would be able to be used in humans as well. This is some work that we've done in bladder with the aromatic amine-forming biphenyl. It's a bladder carcinogen. You can see we're getting high recovery of the DNA adduct from FFP tissues as well. We have some limited data on PhIP as well in the prostate tissues. So a large number of the specimens we can get back DNA from FFP tissues. So this is a major untapped resource that we believe can be used in molecular epidemiology studies designed to assess the role of chemical exposures and cancer risk. Lastly, I'm going to do this really quick because time's running out. We've been targeting for DNA adducts, but we know that there are many chemicals in the diet and environments that contribute to DNA damage and possible cancer risk, and we're developing mass spectrometry methods to look for these different types of adducts simultaneously, not just to target on 1 or 2 adducts. We know that DNA damage can occur at multiple sites within the 4 DNA bases. Our methodology, again, by ion trap mass spectrometry, we do data-dependent MS to lose the deoxyribose, and we can instruct the mass spectrometer, it's a smart instrument, to screen for all chemicals that putatively lose this 116 and then do MS to the 3rd and try to see whether we can identify adducts. And we did this work with human hepatocytes. We treated untreated human hepatocytes, we treated them with the carcinogen forming biphenyl. And again, as the analytes go into the mass spectrometer, it does tandem mass spec on the top 5, top 10 ions. And then lose 116, it will do MS to the 3rd. So this is our control. And this is our ABP treated sample. What you can see is that there were 3 peaks that the instrument saw that lost 116. They all successfully went underwent MS to the third. We got high quality mass spectra. I expected to see this adduct. We knew that this was the major adduct formed in animal models, and it's been reported in humans. I didn't know that we had these adducts. I don't have standards for them, but the instrument picked these out. I went back to the literature and saw that these two adducts were reported over 30 years ago in vitro reactions with cath-thymus DNA. This technique, it's a wonderful technique, but it's biased for adducts present in high abundance. This is the cartoon that shows this. Again, the top 5 ions go into MS. What we've done is develop a new technology to screen for everything at once. Everything gets broken apart, and retrospectively, after the run is done, you can do post-data analysis, and you can look for the precursor ion. You ask the question if anything got fragmented to lose 116, and if these two peaks coalesce, this suggests there's DNA adduct. We've done some spiking experiments with cath-thymus DNA with this adduct and shows that it works nicely. We went back to some of our PhIP positive specimens and negative specimens, and we showed that this works. Similarly for Aristolochic acid, it also works. We're quite excited about this. So all the data is in these files, so we can just retrospectively go back and type in any sort of putative adduct and see whether it's being picked up by our methodology. I'm oversimplifying it. We still need to refine and improve the method, but this is where we're trying to go with this technology. It's also very challenging because we're literally looking for needles in a haystack, and if we want to have an algorithm that can automatically detect these adducts, you want to be able to look for the precursor ions as well as its N+1 and N+2 isotopes to have the molecular formula envelope. And this is work that's ongoing right now with my colleague Scott Walmsley, who's a brilliant bioinformaticist. I don't have time to go into the algorithms that he's developing, but I want to cite his name because he's doing some exciting work in this area to automatically detect adducts. And we've been looking at some of these renal tissues from subjects in Taiwan. And this is actually with his algorithm using PLSDA techniques. He has separated out the smokers, which are in blue, from the non-smokers. We don't actually, most of these adducts, other than Aristolochic acid, we don't know what they are yet. And these are putative candidate biomarkers. Again, this is only N is equal to 3. We have many more subjects, smokers and non-smokers, to see whether they'll separate out. The algorithm is under refinement to improve the approach, but it appears that we're going to have many adducts that we don't know what the structures are. And perhaps not surprising, metabolomics doesn't know either. And so this is where the bulk of the work is going to be. So in conclusion, it's incredibly important to understand the biochemistry of genotoxicants, how they undergo metabolism to develop robust biomarkers. In humans, some chemical biomarkers can be used to assess exposures. Others may reflect biological effects. I want to emphasize each carcinogen, each genotoxicant has its own unique challenges in developing biomarkers. There's not one single endpoint that's going to fit all. Hair and FFPE tissues are untapped biospecimens in environmental exposure and carcinogen assessment that I believe can and should be used in future molecular epidemiology studies. We're excited about the DNA omic technologies that we're developing. And ultimately, our goal is to link some of these adducts to mutational spectra and cancer risk that will help us advise the public to mitigate certain types of exposures and hopefully decrease cancer incidence. So I've been very fortunate to have a wonderful group of collaborators in my laboratory. B.H. Young, who developed the FFPE work, Jingshu Go, the DNA adductomics, Majda on the cell work and prostate, Scott on the bioinformatics approaches and the algorithms, and of course the mass spectrometry analytical biochemistry shared resource, now led by Pete Vialta, who's been an incredible collaborator. I gratefully acknowledge all their excellent work, and Chris Waite and Perry Murrigan here in the Departments of Urology and Medicine Pathology, the urology staff, the Bionet group, Roman Lab in Stony Brook, and Dr. Chen in Taiwan, Ghislaine Sciello at ILARC, and most importantly, the patients who have gratefully allowed us to use, of course, their de-identified biospecimens to advance this research and our funding through NIH. And I'd like to also cite the Masonic Chair in Cancer Causation that supports some of our research. So thank you very much for your time, and I'd be happy to answer any questions.
Silly question from a guy coming from an awesome beef-eating tradition country. PhIP generation depends on grilling method, i.e. high temp short time versus lower temp longer time, or that heat source propane versus charcoal, wood, etc. And what about the quality of meat, feedlots versus grass feed?
Good question. These compounds, the formation of these compounds are driven by high temperature and time of cooking. The longer you cook at high temperature, the more well done your meat is, the higher levels of these carcinogens are going to form. Different types of heating elements are going to produce different levels of these carcinogens. They will form in grilling. They will form under barbecue gas or charcoal. I quite frankly, I don't know, and I should know this, but it means that it's probably not out there. In the literature, there have been no quantitative measurements for the carcinogens. For instance, grilling a burger on a pan versus broiling it versus barbecuing it. I don't know of any data that's quantitatively measured them. Lower temperature will not produce these compounds. You really need to be above 150, 175 C. And again, it's time dependent. So if there's a take-home message, I tell to the lay people, don't burn your meat. All right, it's the burned surface portions that contain these compounds.
PhIP adduct levels do not correlate with 1A1. Have you done a GWAS study to determine which genes are responsible for variability?
Well, first of all, it's 1A2, it's not 1A1. That's the primary activator. We had hoped to do that, but unfortunately the problem is the levels of adducts that we have, the numbers are just in this cohort, just not high enough for us to get reliable biostatistics. We're having less than 10% positivity, unfortunately. We'd like to go back into other cohorts, for instance, have a cohort which is higher levels of African Americans, which we believe are going to be exposed to high levels of this compound, and thus we may have higher levels of adducts, and we would very much like to try to do those types of GWAS correlations. It's just a question of numbers.
Is it known if PhIP modulates AR transcriptional activity? You also mentioned PhIP having effects on ER. Is that binding to ER, or does PhIP modulate ER activity?
I haven't done the work on the estrogen receptor. The data, there's docking data, there are in silico studies that indicate that it binds with high affinity, and there is some data in cell lines also supporting that. What we would like to do if we get refunded is we will be looking at the potential of PhIP in prostate cell lines that do express the androgen receptor, as well as those that don't, in order to understand some of the non-genotoxic effects, including cell proliferation and oxidative stress that may occur. As far as the estrogen receptor, it's been reported to bind to the estrogen receptor at extremely low concentrations. I believe nanomolar or even less. So we're very curious to see what it may be doing with the androgen receptor. But there's limited data in the literature.
Any racial or ethnic differences in PhIP generation?
Well, it depends on how they cook their meat. So for instance, African Americans have higher incidence of prostate cancer. I think it's about, what, 1.8-fold higher than Caucasians. And we know that, this is anecdotal because there's not enough data in the literature, we do know that African Americans, there are a couple of cohort studies out there, they have higher levels of urinary PhIP biomarkers, and so they're exposed to higher levels of this carcinogen than Caucasians. Interestingly, in our data, we have to stratify it out, and we'll be working with a biostatistician to do that. We don't have a very high percentage of African Americans in our cohort here in Minneapolis. However, in our hands, or in our data set, clearly the African Americans have higher levels of PhIP in their hair than the Caucasian counterparts. We have normalized for melanin content, but I can't exclude that there aren't potential other factors. The structure of the African American hair is somewhat different from Caucasians, and the melanin content is often much greater, whether that we're compensating for that by measuring normalized epinephrine. I'm not sure yet, but our data does also support that these African Americans have higher exposure to PhIP in hair. So it could be that, again, diet could contribute, and then there could be disparities in the African American group, maybe at higher incidence. We'd like to go to other cohorts where there is a higher amount of African Americans to further pursue this observation.
Is there a database available for DNA adductomics?
We were awarded an R03. I think I mentioned this to you, and myself, along with Jingshu Go, one of my senior staff scientists, with Scott Wamsley and Pete Vialta, in fact, we have a small communication and talks watch in chem research and toxicology that will be coming out this month, describing what we're doing. We have assembled a collaboration of about 38 investigators worldwide who are donating DNA adducts to us to characterize by MS to the second and MS to the third spectra. The libraries of these adducts will be presented online, publicly available for free. They'll be with MONA, they'll be with HMDB, and we will also be populating these in PubMed. And so we hope to have at least 500 adducts as part of the library. Of course, there may be many unknowns that none of us are working on, so that's another challenge. But this is a start, and this is something that we are doing currently, and we'll be publishing these data and populating the databases in the next year or two.
Regarding PhIP in prostate cancer, it binds AR. Is there evidence that it actually is an agonist of the AR? Also, this would suggest that it is a tumor promoter and not necessarily a true carcinogen, correct?
No, incorrect. It may be acting by both. I don't know if it's an agonist yet or not. We need to do those studies. I don't think that's been reported in the literature. That is very preliminary. We believe, and it's a question of the dose, okay? At high levels in the cancerous cell lines that we're using, it is genotoxic. It does form DNA adducts, so it is acting as an initiator, possibly, if during cell division that adduct induces a mutation. We've also seen that it is inducing oxidative stress. We have shown that we are decreasing levels of glutathione in the cells. We are looking at other oxidative markers of oxidative stress, which is ongoing. But those levels that we're using, at least in this cell line, are higher than one would probably expect in vivo. So this is very interesting data mechanistically. Whether it applies in humans, we're not sure yet. We will be doing further studies in other cell lines, as well as in peripheral zone prostate epithelial cells that are normal, to see what types of sensitivities and responses are. So it's acting both as a genotoxic agent and a tumor promoter, but it's a question of the dose, and we need to better define that.
LNCAP, LNCAP, is a cell line derived from a prostate cancer lymph node metastasis. In your functional studies, have you considered or tested differential effects of PhIP in models that represent earlier stages of prostate cancer or normal prostate cells?
It's a good question. That's part of our grant renewal, and we have not done that. We've started with this cell line because they were highly responsive towards PhIP and formed adducts at super low levels. But yes, we plan to do that in the future if we are refunded.
Well, excellent. I'd like to thank everyone for joining us today, and thank you to Dr. Turesky.